Glioblastoma is a devastating disease with median overall survival of roughly 15 months. We are in need of new and effective therapies. However, there are a number of barriers to effective GBM treatment. First, the blood-brain barrier limits transport of molecules from the blood and into the brain, which is especially relevant at the invasive edges of tumors. The blood-tumor barrier refers to high interstitial fluid pressure within the tumors, limiting transport of molecules from the blood and into the brain tumors. Here we're using MR-guided focus ultrasound and microbubbles to overcome these barriers for delivery of our therapy. Secondly, the tumor ECM is dense and electrostatically charged, hindering diffusion of molecules within the brain tissue. For this reason, we're using brain-penetrating nanoparticles made by our collaborators at Johns Hopkins University to increase distribution of our therapeutic. These BPNs are made of a densely pegylated polyethylenamine, which enhances diffusion in brain tissue. To the right here, you can see 3 different formulations of PEI nanoparticles that were conjugated to a GFP plasmid and directly injected into rat brains. And you can see in the red box here, these densely pegylated brain-penetrating nanoparticles are able to transfect a much larger area compared to the unpegylated nanoparticles and conventionally pegylated nanoparticles. Lastly, GBM tumor cells themselves are genetically complex with mutations in multiple signaling pathways. To overcome this obstacle, we are delivering a microRNA gene, specifically tumor-suppressive miRNA-34a, which has oncogenic protein targets in all 3 core GBM signaling pathways found to be deregulated in the 2008 Cancer Genome Atlas study. miR34a also shows decreased expression in human glioma specimens, and when overexpressed in vitro, leads to decreased proliferation and increased apoptosis in glioma cells and glioma stem cells. Here, we have delivered these miR34a BPNs in a U87 intracranial xenograft model 5 days following tumor implantation. We performed T1-weighted contrast-enhanced MR imaging to visualize the tumors, which can be seen in these pre-FUS images. And the tumors are indicated by the yellow arrow. We then intravenously injected microbubbles and the miR34a BPNs and applied focus ultrasound at either 0.4 megapascals or 0.5 megapascals. We then performed the same MR imaging to obtain the post-focus ultrasound images, which can be seen on the right here. We see a larger area of enhancement as well as an increased pixel intensity in the enhancing region in the post-focus ultrasound imaging. Also, we compared the post-FUS images to the pre-FUS images to obtain pixel intensity fold changes at each of the pressures and found an increase in pixel intensity fold change at 0.5 megapascals, indicating enhanced gadolinium leakage at the higher FUS pressure. To assess tumor size, we performed T1-weighted contrast-enhanced imaging on a 7 Tesla MRI scanner at 2 post-treatment time points, day 12 and day 20 post-tumor implantation, which correspond to day 7 and day 15 post-treatment. Here, you can see representative images from each of our 4 treatment groups in this study, with the tumors indicated by the yellow arrows. We use these images to quantify tumor volumes within each of the 4 groups. The gray bars, the dark gray bars, all the way to the left, show tumor volumes for completely untreated animals. And so this represents the baseline tumor growth for our xenograft model. The light gray bars show mice that received only an intravenous injection of the miR34a BPNs, representing effect of the BPNs from EPR effect alone. The green bars show mice receiving the miR34a BPNs and 0.4 megapascals focused ultrasound, and we didn't see a difference between this delivery group and the EPR effect alone. However, the blue bars show mice receiving intravenous miR34a BPNs at 0.5 megapascals focused ultrasound, and at day 20 or day 15 post-treatment, we see a statistically significant decrease in tumor size. In conclusion, focused ultrasound and microbubble delivery of miR34a BPNs can lead to decreased tumor volume in a mouse glioma model when delivered at 0.5 megapascals. We're currently working on delivery of a microRNA scramble BPN as an additional negative control to help differentiate between effects of the delivery procedure alone as well as from overexpression of the microRNA. Additionally, we're analyzing microRNA expression levels and protein levels of miR34a targets within the tumors and evaluating proliferation and apoptosis in tumor sections, and we're also planning to test this in a glioma stem cell model. I would like to acknowledge my collaborators at UVA and Johns Hopkins. Thank you.